everybody. So glad to have you all here. My understanding is what we're going to be doing with this particular session is just asking questions. And so I was just counting up here. We've got 1, 2, 3, 4, 5, 6, 7, 8. 8 physicians, PhDs, whatever you want to call us, clinicians, researchers, etc. And if you went to go to your specialist and how much that appointment costs, and we have times 7 up here, it still is less than the plumber would cost, I can promise you that, or your cable repairman coming out. But in all seriousness, this is a great and probably unique opportunity to ask literally whatever question is on your mind. And so I would like to start off, and if you raise your hand, JR can't see it, but some of us can, and we'll come out and grab, and you can have a microphone. But I would like to ask an initial question. What is the importance, if someone just gets a diagnosis, or maybe they've had a diagnosis of choroideremia, what is the importance of seeing a retinal specialist on a routine basis? Let's just start with that question. When should that happen?
Yeah, so this question came up in the What to Expect at the Ophthalmology Visit Forum, and I look forward to what Ian has to say to it. But I think there's all kinds of really capable, skilled eye doctors, realizing that many people may live somewhere where going to a retina specialist, going to a inherited retinal disorder specialist requires a lot of time to get there, right? So I'm cognizant of that. I do think it's worthwhile to have retinal pictures taken periodically. Sometimes you might, in retrospect, wish you had had those at some point in the future. They're good in the here and now. It's just understanding the lay of the land. I think the other benefit of periodically seeing a retina specialist, or maybe even more so somebody with more focused inherited retinal disorder expertise, is they will be able to tell you more than anyone else what's going on with trials, whether it is a resurgence of gene therapy trials, whether it's what's happening with optogenetics or the photo switch you heard about yesterday. So that kind of information will be harder to come by outside of a specialty.
Try this. Agree with Ted. I think as a patient, I want to see my eye doctor every year because I have not me personally, but I would think that I would want to see a doctor to make sure that everything else is okay. And I agree that imaging is very important. In the past, we didn't have imaging as we have today, where you can use this OCT to get a map of the back of the eye. And there are some cases, it comes to mind, that there are some cases where there is what we call a neovascular membrane that can occur underneath the retina that needs to be treated. Don't wait. It needs to be treated. You've heard of age-related macular degeneration and the wet form of neovascularization that requires injections in the eye. And there are some cases of choroideremia that require injections because a neovascular membrane forms. And how do you find that? Well, you would probably find that using your OCT. And if you were a patient, you would probably be experiencing some change of vision, let's say in one eye, and that would bring you to something that you would want to keep an eye on. And there are other things. Is your prescription okay? Is the blurred vision that I have, is it okay? Something I have to live with, or change my prescription? Is the pressure of the eye okay? Is there a cataract forming that I need to be aware of? So various reasons, and then you would be able to also ask the questions that you have.
Thank you. Thank you. Any other thoughts from any other panelists before we continue to open up for some other questions? Questions from the audience. This is it, guys. This is you. Hey, Mike, right in front of you here. We got you. You bet. What? Why don't we just go down the line and just if you would say who you are and where you're from and your area of specialty, and we're going to start from our right and we'll move down to the left.
Hi, I'm Malia Edwards. I'm a PhD scientist at the Wilmer Eye Institute at Huckfeldt.
John Lueck. I'm a PhD researcher. My lab works on nonsense mutation.
Katie Bales, research investigator at the Atlanta VA and Emory University, and I'm also within the Center of Visual Neurocognitive Rehabilitation, and I specialize in neuroprotective methods against retinal degenerations.
Rachel Huckfeldt. I am a clinician scientist with a retinal dystrophy-focused clinical practice at Mass Eye in Ear, where I've also run our involvement in the Spark and Biogen choroideremia gene therapy trials.
Hi, I'm Ian MacDonald. I'm a medical geneticist, general ophthalmologist, and I'm at the University of Montreal.
Joe Porter. I'm a basic researcher in nonsense therapies at the University of Rochester in John Lueck's lab.
My name is J.R. Rizzo. I know many in the room. Great to see you all here again. And I am a physician scientist and leader at NYU Grossman School of Medicine, principally trained as a rehabilitation doctor. And our lab investigates, as many of you know, assistive technology development and testing. We do some work in motor control and also in disability health services.
I'm Jess Thompson, cardiothoracic surgeon in Arizona. I'm the chair of the research committee and work with the Scientific Advisory Board. Great to be with you all. And do we have some more questions? Eric, did you have a question? Okay, right behind you, Eric.
When you were just talking about the need to go see a retina specialist, my thoughts go to what many of us as patients go through when there is the potential of participating in a trial. And I'm wondering if there's also real value in being able to go to a retina specialist and get the OCTs on, I don't know how regular, every other year or anything like that, so that we as patients might even be able to build up our own natural history data that we could in turn share if we're looking towards trials or to show long-term progression of the disease.
I agree with that.
I do too. Any tempering of my comments about how often to be seen is me holding back my inclination to have as much data as I can on anybody.
Just to follow up on It's a quick follow-up on that. How easy is it for a patient to ask for all these documents from their doctor?
I think we can do a better job of identifying ourselves as we talk. So this is Rachel Huckfeldt answering the question. Sorry about that before. To the question, it is frankly kind of a pain at my hospital. It's not that we don't want to give you the data, it's that you have to submit a request to medical records to get your images on the disk. So it can be done, it just always seems to me that it's a little too hard. As an intermediate, I often have people take pictures of their pictures when they're in the room with me.
Hi, good afternoon. This is for Dr. Rizzo, and other people in the room might know the answer to this, but I don't. What is a smart cane, and what is the value of it, and the difference between a regular cane?
J.R. Rizzo here. Great question. So usually a smart cane, I'm not sure if you, I was going to reference my talk, but let me throw that out in case folks missed it. By the way, best talk of the day. I'm an aside. I'm a little biased, but anyway, smart cane is usually when you add electronics to a cane, right? So some form of sensor technology, and usually those sensors are being used for what we call distance and ranging information. So a lot of times in conventional use and practice, you'll see ultrasound sensors similar to the little parking sensors you'll see on the front bumpers of most cars these days. Same sort of thing. And so usually those are emitting ultrasonic pulses that's bouncing off of ambient objects. We're coming with that information, or that energy, electromagnetic information is coming back to that sensor from a receiving standpoint, and then we can gauge depth. That's being communicated typically through tactile or vibratory sensors in the handle of the cane. Now, I'll tell you, in our practice, we get asked about questions in smart canes, and I'm curious to see the thoughts from the rest of the panel, but by and large, having relationships with a lot of very senior orientation and mobility instructors, a lot of them will tell you that it's very difficult to leverage that information because, as you know, if you've been trained in white cane technique, we're constantly swinging this cane back and forth, right? So you have these sensors and you're constantly swinging them back dynamically. And we know from other studies that we variably swing the cane at different rates and different arcs. Great work from a guy named Rob Emerson at Western Michigan. And so what that means is you're never consistently getting that feedback. And so I would think twice before we use smart canes. And they are evolving and rapidly evolving. There are a couple, sort of that take the majority of the marketplace, and I don't want to get into specific ones and any details, but I would be mindful, and I would work with your local orientation and mobility instructor. But let me leave it there. I don't know if anyone else on the panel wants to add. I don't believe there is one here. I don't know. I haven't been in the exhibition room. I don't know if Eric or Corey or Kathy knows, but no, there's not. Okay. That's what I thought. Good question.
Hey guys, it's Corey MacDonald. And not really a question, but more of a comment following up on reasons to see a retinal specialist. For those who participated in either the SPARC or Nightstar/Biogen trials, another very compelling reason is to keep your data current. Because these things are not done yet, and that data could be really important moving forward as we try to get this thing pushed to the finish line.
Hi, Vince Taldone from New York. I had asked this question of Dr. Alamon earlier. I think the transmission problems really messed up the problem. He didn't answer it, but I know he would have if he understood what I was asking. There are trials, particularly we heard yesterday from Ray Therapeutics. There are others working to flip that switch, cause cells that aren't photoreceptors to be photoreceptive. That's very exciting for folks with advanced choroideremia like me. But for those of us who still have those tiny islands of functional cells, we have to worry about what does this treatment do to those cells. And of course, the companies doing the trials really don't want to say much. Their employees can't say anything under their NDAs. What are your thoughts, or what have you heard about the impact on the still healthy photoreceptor cells?
Okay, it's Katie Bales. I am not a geneticist. But just as an overall observation, anytime you make an injection to the eye, that's going to cause inflammation, right? It's going to cause an inflammatory response. Doesn't mean that that's just a risk or weighing out pros and cons with these sorts of vectors and these AAVs. So yeah, just keeping that in mind. And so I will say that there has to be some sort of there have to be cells present in order for it to work, right? To be able to reprogram and to be able to infiltrate into the cells and to reprogram them. So just coming from it from an inflammatory response aspect, I would say a con of these injections and is that it will induce an inflammatory response. It doesn't mean it'll break everything like a huge cascade of inflammation, but it will cause an inflammatory response. So just keeping that in mind when you do these injections.
So I'll add to that. This is Malia Edwards again. I'm not a clinician. I'm a basic scientist, but I have looked at a couple of eyes with choroideremia histopathologically, so looking at it in the lab. And I would say, and just by what we know, what you can see clinically, there's a lot of changes that occur in the retina that, I just turned on. My concern would be, again, that inflammation, and also it's not going to prevent the further damage that's already occurring in the eye. So I think you're going to have a continuation of cell loss, and as choroideremia progresses, when we looked at very late stage diseases, and I think Ian has also looked at these, so he can, Dr. Mack, can comment on this as well, but we don't see, I don't know how many ganglion cells are going to be left. We have changes to the blood vessels that within the retina that you need for even the ganglion cells to survive. So I think a lot of the changes that are happening in the retina, and you have, going back to the inflammation, the cells that are there are kind of primed to be targeting things that shouldn't be there because they're kind of angry because of everything that's happening in the retina. So they may not respond. I don't know how they're going to respond, and that may increase the inflammatory response. So I would kind of agree that would be my caution, and a continued, injection every month or so, I think is what was said, or every 6 weeks, wouldn't necessarily be fun, I don't think, for the patient. But I don't know if anyone down there has comments.
This is Rachel, and I'm gonna break your question into like two kinds of risk, right? What is the risk to you right now? And I'm going to take that at a very visual level, right? You have an island of vision that you use, and how will that vision be affected if there's now this additive therapy on top of it? And I'm going to answer that first from the perspective of like nothing goes wrong, and that new therapy maybe even works. We don't know, right? We don't know what it's like to have the island of vision and then have something different than regular vision around it, right? So we don't have an answer to that one at this point. But then I think there's also the kind of risk that Katie mentioned. And we know that, I think the Kiora, I wasn't here yesterday, but I think the Kiora photo switch you heard about, that's not viral based, but we know for sure that AAV based therapies, like any genetic therapy, almost any genetic therapy uses, are inflammatory, right? So there's always risk that comes with that. But then the second category of risk is, and anytime I'm talking to anybody about a clinical trial, we talk about this. What is the risk that participating in this now will close off options to you later, right? And that's where, for anything viral mediated, whether optogenetics or anything, we don't know how safe it is to put a second virus into an eye that's already seen one, right? In terms of suppressing an immune response. You're not asking about subretinal therapies, but when we do a subretinal therapy, that's another question. We don't know if we can go back to that eye and deliver a second one. So depending on the therapies, there's all kinds of sort of more future-oriented questions that are unanswerable right now, right? But people just have to think about them and know their comfort level with them.
Ian MacDonald, thanks very much, Rachel, for your comments, and everyone else. I wanted to perhaps add a little bit of a discussion around clinical trials and their phases. So some of these are in phase 1. This is a safety trial. Some of them are in phase 2. This is a trial that is trying to show that it's actually effective. And then phase 3 is where it's a broader number of patients. And some of these phase 1 trials include an additional amount of a dose of, let's say, the viral virus with the vector. And so it's called phase 1B or something like that. I get a little bit mixed up. You're trying to see if there's a difference in between one dose and another. And I'm not sure it's by design, but you can imagine that most of these trials are treating individuals who are over the age of 18. And in many of the trials, the patients are being treated, they're significantly older than age 18. And for choroideremia, certainly by the age of 35, there's been significant loss of retinal pigment epithelium. And so part of the question one of the questions that clinician scientists have is that, why are we treating at such a late stage? Why aren't we treating at an earlier stage? So I think the earlier we treat, more potential to prevent decline in vision, perhaps the more opportunity for benefit. Now, if we switch all that discussion I've just been mentioning about to the optogenetic approach, these are patients who are being treated because they have very limited remaining visual function. It may be light perception or worse. And many of these patients have retinitis pigmentosa. And yes, it's perhaps nice to think of I get a little bit mixed up here because I'm not a Catholic, but Catholic means that you're able to include everybody. And I think there's what's the other? Yeah, gene agnostic. So I know there's there's atheists and there's agnostics and it's what you believe in, and it's how that's right. It's what you believe in, and so an agnostic treatment with a virus that's carrying opsin gene and making the remaining elements within the retina photosensitive. That's a great idea. But when do you apply that? And if you apply it to end-stage retinitis pigmentosa, is it fair to say, well, we can use that for end-stage choroideremia? Why don't we try that? And unfortunately, not unfortunately, but it might work. So fortunately, it might work, but it might also not work. And then how do we decide it's successful? This is where we're really struggling, both patients, families, and clinicians. We had great hopes for this, really did. We were so committed to it. Did we pick the right group to treat? Did we pick the right time to intervene? All those questions are still present.
So we have a couple more questions, but just as a follow-up, because it seems like we're talking about clinical trials. It seems like a lot of the clinical trials, if you've been in one before, you can't be in another one. So if I've got a child with choroideremia, how do I decide if there's, because it looks like there's a lot of clinical trials hopefully that are coming down the pipe, how do I decide which clinical trial to put my child into, or any? That's an easy question, guys. I'm talking to you.
So easy. This is Rachel
In two
again.
words or less, I would like an answer.
I'm going to make it easy for myself and focus on children to start with. And you can maybe generalize for yourselves from there. I think one thing is for everybody, eligibility criteria are going to be a major factor that will tell you whether something is an option for you or not. And age is part of that. I think, this goes back to what Ian said about when you enter a trial, will determine how much baseline information there is about safety versus and maybe efficacy. So often by the end of the initial phase 1-2, we have a pretty good sense of safety. We might, for some therapies, have a whiff of a sense for efficacy. And some people just don't want to join a trial until that information is available, right? Everybody's thermostat on their setting of their ability to tolerate risk, and we're pretty clear to you when we're talking to you in a trial setting what the risks are versus no guarantee of benefit, right? People's thermostats are set differently there. I think this comes back, though, Jess, also to the question tangentially to the question you started with, which is like, how often should you see a retina specialist? Well, it's pretty nice to have a relationship with somebody that you trust when these questions about trials come up, because you can be sure that you will get, an opinion. Opinion's not fact, right? But you can get opinion and information from someone that you've hopefully come to trust over time.
Hi, I'm Carol Brennan. My son Sean has choroideremia. He's usually at these conventions, couldn't come because of other things going on. So I texted him and said, do you have any questions if I get a chance to do this? So he texted me these two questions, which I think are unanswerable, but I'm going to throw them out there anyway. Are any of the treatments being worked on likely to restore lost vision in the next 10 to 20 years? And what are the projections about how soon gene therapy will be approved by the FDA?
Good question. Again, very easy questions to answer.
So Ian MacDonald, very good questions. We're hoping that in the area that's adjacent, Dr. McLaren talked about the smooth zone and the area of the retinal pigment epithelium that is seemingly intact. But then there's a border area around the smooth zone. And could that border area that is marbled, let's say, could that be rescued? And could that improve not so much visual acuity, but maybe visual field? We don't know. From my perspective, I haven't seen that, but it might be possible with the gene therapy. What I think would be expected from a gene therapy, would hope for, is that visual function, both visual field and visual acuity, remains stable, and that would be a significant win. Second question is a $68,000 question. It used to be $68,000. I don't know why, but it's $1 million question. I don't know where the FDA is in some of these things, but it's a conversation that occurs in between the sponsor and FDA with significant discussion back and forth as to whether it's approved. For Luxturna, that took a long time for the product that is treating Leber's congenital amaurosis in not only the adult population but the pediatric population. I better stop.
Ian never needs to stop because he always says good things. But this is Rachel again. I'm just going to speak to the second question about the FDA, maybe in a different way than your son was asking. And that is, ophthalmology trials and products in the FDA and any guidelines we have, aside from making sure something is safe and effective, they are really not directed towards therapies that treat choroideremia or conditions like RP or slowly progressive genetic conditions. And so, for example, if you have a ophthalmic therapy that restores 3 lines of vision, well, that's a winner. We heard from Dr. Alamon this morning about the challenge of doing that in choroideremia, right? And the flip side is if you wait for somebody's vision, if your control group is waiting for somebody's vision to decline by 15 letters, like that's just not a tenable length for a clinical trial. And so this in general just speaks to the need for better endpoints for retinal dystrophy clinical trials. And that's not unique to choroideremia. That's something that the Foundation Fighting Blindness is working a lot on. I chair our endpoints working group for the FFB Clinical Consortium. We are just one of many entities in the field right now thinking a lot about endpoints. So I think that is where hopefully our collective voice can help make sure, our collective efforts can help make sure that when there are therapies for choroideremia that come into clinical trials, maybe we have some better ways to measure outcomes in the future that just help us, guarantee that we are able to identify effective therapies and hopefully get them approved.
Katie, yesterday afternoon you did the health and benefit, which was great. I appreciated hearing all that. And you mentioned how the NEI is being impacted now in funding. And again, Rachel, you just talked about the FDA. Is that all legislative? I mean, what do we do? It seems like, are those people who are qualified to make decisions? I mean, I know that sounds stupid.
No. It's not stupid. It's complicated, right? And thank you for your question. And I think it is loaded, especially all of us sitting up here. It just feels, anyways. We are facing some challenges with every new administration. There's going to be changes, right? But these are some really drastic changes we've never seen before. I mean, as far as across the board, getting the indirect cost alone being cut to 15%. If you're not aware of what indirect costs are, they're just built-in funds for grants, such as, we through the NIH that help keep the lights on. Literally, we're paying people for the administration, keeping the lights on in the buildings, helping pay for people who help perform the research as well, like core facilities that are essential for all of our research entities. And so, yeah, there are some drastic cuts, and study sections just started meeting again. So that means that all these grants that were submitted, finally are getting reviewed. But I mean, there are some major financial cuts. So a lot of researchers like ourselves may or may not get funded. And this is this is what we dedicated our lives to. Yeah, it's a nightmare. But we're going to keep going. And that's why we're all here today. And that's why we're so grateful to also have people, this wonderful foundation of all of you, because you're so passionate. And that helps drive us to keep going and to face all of these drastic, awful changes that have been happening and will continue maybe happening for the next 4 years that we're going to have to face. But either way, we're going to keep pushing on and get to the bench and keep working. Yeah.
Add to that, so like really the only way you can change this now is to call your congressperson. What day is it? Today, because the bill is being pushed to be passed on Sunday. And so if this doesn't happen, what's in the bill is not good for not only research is important to this group, but any group of people affected by the disease.
And I'll add one more thing to that. With the restructuring, they're actually trying to do away with the NEI, and that's kind of what I talked about yesterday. Was keep the I in NEI because they're trying to reorganize and reabsorb the NEI into a larger, like neuroscience division, and so that means we're going to get wrapped up, and our grants that are specifically eye related will be wrapped up with other grants that are related to Alzheimer's and Parkinson's and other neurodegenerative diseases. And so that'll put us on the back burner. And so please, contact your Congress people. I'm happy to provide. I have some links I showed yesterday in my slide deck. Happy to provide those to Kathy so she can disseminate them out to the group.
One more comment. In that the investment in the NIH is the best investment the United States has ever made, purely on money. And so if you want to make an argument to people, return on investment is about 10 to 1. So if anyone likes money, that's a really good investment.
I just want to speak to the FDA piece. Challenges with ophthalmology endpoints, they go way back, and I think it really speaks to it being relatively new that we even get to test treatments for things like choroideremia, and it is, I think it's an education process for regulators here and in Europe, and I think this is where patient voice is really, really important. I think it was only like 2 or 3 years ago that I heard said out loud at a meeting that it would be, we all want to improve people's vision, but if we can stabilize, that would be a worthy outcome too. And that is a new concept in some ways for regulators to hear, right? So patient voice in all regards is really valuable with regard to the FDA.
And Dr. Bales, correct me if I'm wrong, but right now it's the bill is at the Senate to be voted on. Is that correct? And so, although the offices in DC are now closed, their emails are still open.
Yes, exactly.
And so if you just go to senate.gov, you can find your senator, 2 senators per state, and you can just email them. They will be checking their email today and tomorrow and Sunday morning before any voting. And so whoever your state senators are, you can clearly go get their emails and just email them a quick, hey, this is what, I'm passionate about this kind of thing. So big beautiful bill or whatever it's that's hard to misplace that one.
If anybody's interested in, so I also do work for the Cystic Fibrosis Foundation, and we were provided a link that you can easily access and send messages to. So if anybody's interested in this, I can get that out to you.
Thank you very much. We have a question right here.
Hi, my name is Marty Rice. I'm from New York. I have a question. First of all, I'm been blind. I'm in the very late stages of the disease, but I've also suffered with insomnia my entire life. And I recently was surprised to read on the Choroideremia Facebook group that the REP1 protein deficiency, which causes the blindness of choroideremia, also can cause abnormal serotonin levels, leading to sleep disturbances. I want to know if anybody has ever heard of that. Is it true? And if so, what do I do? Thank you.
This is Katie Bales. I do not know the answer directly to this, but I will say that with blinding diseases, there will be disruptions in your circadian rhythm. We know that. And so that, as far as this serotonin levels, I had not heard of that, but we can definitely get back to you on that question. Happy to do that. And we'll get back to you on that. But going back to, any ocular diseases, there are, with vision loss, you will have circadian rhythm disruptions, and that's exactly what helps regulate your body to say it's time to go to bed. So yeah, that makes sense, and we'll look into that for you for sure.
Where are they at in trying to bioengineer this REB1 protein that's needed for the disease? Is there anything worldwide that is being done on? I kind of compare it to something like hydroxide, like thyroid hormone. Is there something being done to genetically get this protein that the eye is not making?
That's a really good question. There's enzyme replacement therapies, it's like what you're saying. With this, I think it would be it's a delivery problem. So the idea of like going into the eye to deliver the protein, proteins are inherently very short-lived, so it's a durability issue. So certainly you can inject protein into the blood or to different tissues, and if you can get it into the cell itself, it should work. But then that would only last for maybe a day, and then you have to do it again. And so it's a great idea, but durability is one of the issues with that approach.
Hi, it's Vince Stalzone again. This is actually a question about pain relief due to eye strain. My stage, I can see each of you. If you, smack your head because you can't believe the stupidity of the question, I'll see it. But if you roll your eyes, I won't. Make sure you just roll your eyes. But I've spent the day, and this is every day of my life, seeing as much as I can. It's straining to see. You're in a deep fog, but I want to see everything I can see. Halfway through the day, I have the worst headaches ever. And there are painkillers. I have a migraine medication I can pop that will knock out the pain. Is there anything you've recommended? Any of your patients have said, I do this and it helps?
So I see a number of patients with different eye diseases, and I'm going to take you into another domain. For example, albinism with nystagmus, and many of the children start the day fresh, rested. By midday, they're pretty much done for the day. Their eyes are moving more rapidly. They have eye strain, eye pain. They have to take a break. They have to take a nap in some cases. And some of them take Tylenol or Advil and to get rid of that. So it's a different disorder, but I don't think it's that dissimilar in the sense that, I'll give you another example. For some of the retinal dystrophies, patients will say that they were out all day in the bright sun, and the next day it's like a hangover. They don't see very well. It's taken a whole evening, whole sleep cycle to get back, but they're not quite back to this to the baseline that they wanted to have for vision, and they have discomfort as well. How to treat it? Probably very much idiosyncratic. Sorry, I didn't say my name again. Ian MacDonald.
Yes, I know your voice now.
Sorry. Oh dear. Is that sufficient? Anybody else would like to jump in?
Great question. This is JR. So, I think part of this has to do with visual hygiene, right? And I've been seeing through pathologic eyes since I can remember. I think it's difficult. It depends on the visual tasks and how you're trying to use your residual vision throughout the day. I do think there are certain practices you can follow and certain adaptations you can make. So, for example, I have different gradient tints that I use constantly. I think be really mindful of your eyewear itself. When you're going outside, you should constantly have UV protection and either transitions, or sometimes I travel with different pairs of sunglasses, and I will use them. Different filters, blue light filters, I think are really important. We have to be mindful how much time we spend on digital devices, which I think can really fatigue the eyes and trigger asthenopia, which is sort of a fancy word that means eye fatigue. And I find that if I'm on screens more, or tablets, depending on the desktops and how much time I'm on my desktop, different things like, wavy lines or photopsias and floaters and those sorts of things start to increase pretty frequently. And then being mindful and taking rest breaks, right? So, there are rules like the 20-20-20 rule. I was talking about this earlier with some members, where every 20 minutes you should give yourself a break, a visual break if you're using residual vision. Whether it's 20 seconds or 30 seconds or a minute or even 5 minutes. And, in my own personal practice, I try to get up out of my seat at least once or twice every hour. Sometimes it's getting a beverage. Sometimes I'll close my eyes for a minute, just different practices so I can get more out of my residual vision. And I still get the bad headaches, but I find that, if I'm careful in using those best practices, I can mitigate those effects. So hopefully that makes sense. We will have an article on visual hygiene that's coming out because I don't think it's discussed enough. So email me, Vince, and I'll get you a copy of that article.
Thank you. Sure.
Hi, my name is Rosalind Lewis, and I have 2 sons with choroideremia. My older son also has some learning disabilities, but I'd like to ask Dr. Rizzo or the rest of the panel if they are aware or knowledgeable about any areas. I'm trying to encourage him to more independent living situations. So I think what I'm just looking for are areas or schools, kind of like the Carroll Center in Alaska. In Anchorage, they had a program for the visually impaired and blind. I'm just kind of searching to see if it would be like a residential type program to teach people, independence and how to utilize different methods to be safe, but to be able to live independently.
This is JR again. I don't know of any centers in that geographic area, but I think if you want to focus on living skills and vision rehabilitation, some of what we discussed this morning, I'm sure you could find some occupational therapists that hopefully focus on low vision. We don't have enough of them. We're trying to increase our capacity at NYU and to help sort of spread the wealth. But I do think you can look into the rehabilitative practices in the area. And if you want to reach out to me, I'm happy to see what currently exists and to see if they have the right qualifications. But I think that would be a really great starting point. And then also potentially traveling to another strong rehabilitation center, whether that's in Boston or whether you're going to New York, and then leveraging existing partners to build some alliances and to see if you can get some better care.
Yeah, there are more, Ian MacDonald again, there are in many areas schools for the blind, but beyond that, going off to college, advocacy is very important, so either through your physician or directly from him to the college saying, I need more time, I need accommodations, I have a disability. And colleges and universities are very accepting of that. And some of them have bursaries as well, and full tuition for individuals with low vision. And it's amazing what can be accomplished. So it's not a. Leave it there. I'll leave it there.
My question is about resources and just general information and getting educated at the layman level. You know, some of these presentations were very, maybe 101-ish, you know, just really helping understand the genetics of this kind of situation. Others were very much scientific. People are at different levels of understanding or interest in the information. So is there a clearinghouse? How can one find information about who's doing research and at what level?
I can answer that one. This is Jess Thompson with the Research Foundation. That is a great question. And that's actually something that we hope to work on. You've seen Mike walking around. He's our new Chief Science Officer. And just would like to introduce him to our community and family, because that is exactly one of the things that Mike is going to be working on. Because we need to do a better job of that as a research foundation. And so we hope to, we're not there yet, but we hope to meet your expectation in the near future.
Our website is a starting point. I think it does a pretty good job now, but I hope to be doing more there and then reaching out through social media as well. But watch the website.
And I could say as a researcher, I get emails from families all the time. And those are the ones that I actually respond to. So if you have questions and you're just on Google or PubMed and you just email researchers, for the most people, you know, it may be a little bit of a delay, but they would be happy to get back to you and talk about any questions you have.
I'll just second that. And as a researcher, yeah, that's something I would be, you know, that's one of the emails we would respond to. So I wouldn't, you know, if you see papers and it doesn't make, you know, obviously the papers aren't written for the patients usually. So if you see a paper and you want us, you know, especially anyone that you've seen here today or, you know, over this week, I would contact us. And, you know, I think most of us would be more than happy to kind of talk to you in layman's terms about what we've done.
I appreciate all of your input, but one of the things that JR talked about, and one of the things that we've all learned, is that we need to protect ourselves from bright light and to have that protection. When we got an OCT, right at the end, is that autofluorescence where suddenly it's going with really bright lights. Has anybody done any studies to see this, how much, because it hurts the hell out of us, and our vision turns red, and I'm just wondering at what point with certain diseases is that detrimental? Because I've had a number of people ask me questions, and I said, I have no idea, I'm assuming not, but when you're doing the imaging of the eye.
Yeah, so this is Katie Bales. It is uncomfortable for you guys. I can't even imagine. There has been extensive studies because it is using a laser, and so there have been extensive studies in creating those devices to ensure that they're safe in the clinic. If you are uncomfortable and it does hurt, definitely let your physician know and say, "Is there any way we could potentially speed up this imaging?" That could be a place to start. But again, those images are your history, right? So. But I definitely think that's worth talking to your doctor and saying, is there any way we can make this more comfortable? For me, it's really causing a lot of discomfort because that's certainly something they want to avoid.
Yeah, and when you mentioned that to your doctors, I have learned, because I do a lot of grand rounds and people are going to come in and turn their light on to see me. I always tell them to please turn it down to the lowest possible level. And
that's great.
And that makes, for me as a patient, it makes a big difference. And I also ask, because it's crazy, sometimes I've gone to a clinic where they immediately shine the light in your eye and then say, all right, now let's do the visual acuity.
Yeah, that's just bad timing. That's just not great.
So this is Rachel, and I went to just be really clear to distinguish what the kind of laser that's used for OCT from autofluorescence. So the OCT, you're fixating on a target, the line goes tick, 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 tick. And then what Eric is referring to is I think the screamingly bright blue light.
At the end.
Yeah. So that is giving us the autofluorescence image.
Okay.
I will say in our clinic at Mass Pioneer, we actually get autofluorescence a different way, using a slightly different wavelength that's a little bit less intense. And that's motivated more by comfort than for concerns about safety. But anyone who doesn't like the blue light can also ask their doctor if there's a different way to get the same kind of image.
Great question. This is Jess here again, Jess Thompson, but just follow up with the previous question. In the near future, we're going to be doing a lot of revamping of what we present to our foundation. And so if you will grab me, grab Mike, grab anybody that you see that's going to be on the research committee. So if you go on the webpage, find us. If you have things that you say, I wish I had this, or when I first started this journey, if I would have known that, those are the details that we need to know. So we want to hear from you. Please tell us your experience and what would have made it better for you, because that's what we're going to try to do. And so please just grab us and start giving us your ideas. Email, call, grab us at this meeting. Myself, anyone, just grab us. That would be very, very helpful. So please.
My email is simple cso@yourchm.org, and I would welcome those kind of comments and feedback and suggestions for what we're trying to pull together in the near term. Thank you. CSO, Chief Scientific Officer, at CureCHM.org.
Other questions? Mike, right here. Oh,
sorry, I had
one here, and then Corey's in the back.
One quick.
Oh, yeah.
Kind of goes back. Hi, Carol Brennan. Goes back to what Sean had asked about FDA approval, and this probably is just my own ignorance and naivete about this, but there's so many studies and trials going on in other countries, Canada or Europe or Australia. Why is it that we have to wait for the FDA to approve things? Do they have similar things going on over in the other countries, or what's the deal? Thank you.
So I think it's important to follow the FDA. There's different endpoints, as we discussed before, and different trials that are being performed. Things are approved in Europe that are not approved in the United States. Some, I would say that overall, I'll give you an example. Adlurin was one that was approved in UK, not approved in the United States, and that's because Adlurin doesn't really work. And so I think you just need to pay attention and talk to your clinicians about what is being approved, what is on track, and what are the outcomes, and is it worthwhile to take it. For the most part, I am a firm believer in the FDA and what they're doing for safety, and I think it's really important to pay attention to that.
This is Rachel again, and I think two things. One is that we are a pretty interconnected global community of people working on these things, and so that means we're also saying the same things to regulators in different countries. I don't think there's one country that has it better than all the others either. And Professor McLaren this morning in his talk, he talked about how you need 3 lines of vision gain in the U.S. versus 2 in Europe, and there's a little bit of things are not carbon copies, right? But they're not far off from each other in terms of regulations in one country versus another. And maybe to the other point, sometimes we are more permissive than other countries. There's a number of drugs that have been approved in the last 2 years for a particular type of age-related macular degeneration that are available in the U.S. and the European regulators said, absolutely no. So it's not always one direction in terms of one being more permissive or not.
Okay, I'm about to ask a question that's been asked many years and see if there's any difference in the answer. Progression rates. Why in the heck do they vary? I mean, I've been working with CHM patients for 30 years now. I've seen people using a cane full-time at the age of 14. I've seen people legally driving at 60. And I even know of a pair of identical twins who progressed at different rates. How in the world is this happening?
I think the short answer is we don't really have a clear understanding of that. And if we did, it's kind of the same as why some female carriers have no phenotype and others have, have the same as males, right? So I think the more we can learn about the disease and the more we can learn about, the pathology and imaging, et cetera, I think hopefully that will help. But as far as I know, I don't think we have a real good understanding of that. It's a $1 million question, another one, I guess.
So in other diseases, they've identified modifier genes. So they're always looking at one, for instance, and they're always looking for mutations there, but all of us carry different variants, and that's why we look different, that's why we look different. And so there are interactions between different genes in our body that can be helpful. And in order to, in general, in order to understand how these interactions work genetically, do what's called GWAS, Gene-Wide Association Study. And so in those, if we're going to be successful, it's thousands of individuals. You're looking for a needle case.
I also think, Corey, this is Rachel, that answering this question speaks to the importance of ongoing, just basic science research into just the pathophysiology of choroideremia. So, I think the answer to your question is in everything that lies downstream of dysfunctional REP1, and we don't understand everything that lies downstream yet. And understanding some of those pathways will, in the not GWAS, maybe amenable population of choroideremia, it will direct our attention as to what do we look at to understand some of this variability even among family members.
This is Malia Edwards again. I'll kind of piggyback off that since I'm a basic scientist, and I think part of it is also probably the secondary changes that occur, maybe not necessarily because of that initial changes to the RTE or the choroid, but it's the downstream effects, and maybe everyone responds differently to that. So the more we can understand about, again, the pathogenesis, we can understand hopefully that progression more and identify. And that's where I think combination therapies are probably going to be what ends up where you have maybe one thing that may be a gene therapy that targets the initial onset of the disease, but then other things that can, anti-inflammatories or things like that, other drugs that can target some of the, what might be secondary effects, but once those secondary effects start happening, they're probably just as important to the disease progression as the initial insult. Once the retina starts getting damaged, as it happens in choroideremia and a lot of other retinal degenerations, age-related macular degeneration, you need to be targeting those things as well as the initial.
And this is Katie Bales. I'll speak to it from a basic scientist level as well. So with retinal degeneration, the retina is essentially trying to save itself. It will reorganize itself. And so that's also something, another layer of I won't say complication, but just differences within individuals. Like we all have different vasculature throughout our whole body. So the fact that, we're also individual, and then on top of the fact that the retina is trying to save itself and reorganize in a way to help the cells that are still there function and get rid of the ones that are not functioning, trying to having inflammatory cells engulf dying cells. And so just the retina, at the end of the day, is trying to save itself. So it will reorganize the best way it can. So that's also some differences there.
Yeah, hi, Jamil here. So my question is related to cell replacement. So for late stage patients, the most promising treatment seems to be cell replacements. In the CRF, we have been following the work that few companies are doing. There are ongoing clinical trials for like RP replacement treatment and photoreceptors as well. There's one company in India, the name is iStem, that just completed phase 1 trial and should start phase 2 also in the US. So it will be patients from the US and India. But it is only, I think, including patients AMD and geographic atrophy maybe. So my question, so this is for our retinal pigment epithelium, and I think Dr. Gang has an ongoing trial for photoreceptor replacement. So my question is, could you expand or elaborate a bit on the challenges for adding choroideremia to one of these clinical trials? Yeah, thank you.
So this is Malia Edwards again, and I'll speak to that. And personally, as someone who studies that remodeling, and as Katie mentioned, that remodeling and the restructuring that happens in the retina. So there are a couple of problems that I personally see with the RPE transplants and even photoreceptors, is that by the time you are transplanting RPE cells, the choroid is also damaged, and the choroid is very degenerated. So you have the issue. The choroid are the blood cells that supply the RPE and give them nutrients. So you have to have a way to back that up. And eventually, I think there are labs working towards doing combination RPE and choroid. You also have the issue that the retina has undergone such remodeling and restructuring, and you have lost photoreceptors. The glial cells, which are the cells that I study, are not in their normal shape, and they're not performing all their normal functions. So you have a question of, is the retina going to be able to support or is the retina going to be able to respond to the RPE? And I'm not saying that I don't think it's going to work, and I think maybe it could preserve some vision. But those are the biggest challenges that I see, that you have that, again, those secondary changes that are already occurring in the retina. Replacing the RPE isn't necessarily going to stop those or revert. We don't know yet if those can be reversed. And if those things can be reversed, then that's fantastic. But as far as I know, we don't have enough evidence from animal models and things to know if that can be reversed.
Hello, my name's Eric. I live in New York. I know this might be seen as like a dumb question, but for me, I'm the first known case in my immediate family diagnosed with choroideremia. My mom didn't know her dad. So my question is, I've seen retina specialists for like the past 8 to 10 years, but all I ask normally is like, oh, how does my vision look? Does it look like it's worsened? Are there any other questions you recommend asking your specialist during each visit?
Thanks for the question. I think the first question I'd ask the patient is, when should I see you?
This is Katie Bales. So we have some clinical trials, some clinical studies at the VA that we're currently performing in veterans with age-related macular degeneration as well as diabetic retinopathy. And so I think one of the questions you could ask your physician is, okay, what beyond the best corrected visual acuity test, the line test, the line chart, what other tests can your clinic perform, such as the electroretrogram? It's a very specialized test, but we can still perform it in patients. We do it routinely. It all comes down to the equipment in your physician's office. So definitely an OCT and an OCTA, which is an OCT with an angiography aspect to it, could be beneficial. So essentially asking, what other test can you guys perform during my visit would be a good question.
This is Rachel. I would take that question and even ask it as, are there other tests right now that would be helpful for me to understand how I experience my vision in day-to-day life? And that may be something the clinician would be able to do. And I would follow that up with maybe one final one. Is there anything happening in the field that you think I should know about? There's also the chance that having been at this meeting or being a member of the CRF, you might have more information on that than them, but still ask the question.
Another question. I think I just heard something we don't put on our webpage is what questions can be asked your retina specialist. So thank you. That's a great question, and I think we should add that for people with new diagnoses.
Hi, Peter Boone. I had a kind of a follow-up to the pilot study of the X inactivation. Thought it was really cool that that study was done, and so curious if you think that looking at the how skewed the X inactivation is in female carriers, if you think that that's maybe something that would be worth doing as maybe a predictive measure of how their disease, if they are affected, might progress, and if so, what's your Price is Right percent of skew that you think would kind of affect it? You had to guess.
So what Peter is referring to is the really wonderful virtual presentation, a recorded presentation that Senna Gosek gave today. Senna is a recently minted PhD who's now a postdoctoral researcher in Melbourne, who did this really phenomenal work, collaborative work looking at X inactivation. And I think, Peter, her kind of parting words were addressing exactly what you asked about, which is if we know that there's certain, can we use the level of X inactivation in carriers to make any prognoses for the future? And I think that's where her work ties in so importantly to what Jasleen talked about, which was natural history in carriers. I'm not sure how much, how many numbers of individuals and how many numbers of visits you need to be certain, confident of your outcome, but I think it's really cool, and I hope it moves forward because it would be nice to have availability to rigorous information like that.
So we have the opportunity to have several basic science researchers on the podium this afternoon, which I think is great. And I would say that most of us in the audience don't interact, or maybe have never interacted with someone that performs basic science research. So a lot of what the Choroideremia Research Foundation does is fund basic science research, which a lot of us don't understand. I'm going to ask you the question, if you could talk to us, which you are doing now, what should we know about basic science research? Not necessarily your specific field, but just in general, why does it take so long? Why is it so expensive? I'm not sure. If we came up and we were just having a cup of coffee, what could you tell me about basic science research? Because we fund a lot of it. What should we know about what you do?
What are we getting for our buck?
Are we getting 10 to 1 like the NIH? Why not?
I'll start off by saying I think basic science research is a misnomer. I think it should be called discovery research. And I think that's really what it is. So there's a lot I can talk about this. But there's so much that we don't know. We don't know what we don't know. And there's a lot of things can be solved by just knowing more, if it's related or not related, perceived to the problem in front of you. And so, I hear a lot of people say, why are we funding research in worms? Why are we funding research in this organism? Why aren't we doing things directed in, the next closest animal model that would have eyes, for instance? It's because a lot of what we know now is helping with how enzymes work, Repliform works, is because they were discovered in these organisms, and they work there. You move your way up. There's just so much to look at, and you don't know if it's going to be important translationally. By translationally, I mean, is it going to turn into a therapeutic in the end? And never, I don't think there ever has been a case where you start off and it's just a linear line and you just work your way up to a therapeutic. The other thing I'd like to point out is that the percentages of therapeutics that exist in the world, I would say that 99% of them started in a discovery research lab. There are very few therapeutics that started in industry, in pharmaceutical companies, and ended in pharmaceutical companies. What they generally do is they take ideas that were founded in discovery labs, and then they put a ton of money into it to make it into something that goes out. And so I have more to say, but that just gets started. Academic basic science then, but I'm trying to change the name.
I guess this is Malia Edwards again. I will just kind of reemphasize that, we do, yes, I agree, it's more discovery, and we're really trying to understand what's happening in this disease. And, even just understanding how the eye works is a lot, involves a lot. And at the same time, we're also developing new scientists in the lab, bringing more people in to do the research. And training young people, people who have just graduated, even undergraduates who are working in the labs to do this. And I think it's, you look at the dollars, and if anyone, you would be shocked at how much things cost in the lab. I mean, I will tell a student, here's 50 microliters of what we call an antibody to look at a specific protein, and that's $600 or $700. And you tell people how much things cost, and immediately students freak out because they're like, wait, you want me to touch this? But all that goes into, again, this discovery that then leads to us understanding the disease process better, and just, the more we understand even just the normal function of the eye, we can understand how cells interact and how different proteins in the eye interact. So that's how we, get to these more translational work.
There's a cool story, you probably are all very familiar with CRISPR, which is gene editing, right? So that actually started by people looking at bacteria. It's how bacteria keep viruses from infecting them. And what was really, how far away is bacteria from saving an infant's life with a gene therapy, right? And that's exactly what happened 2 weeks ago. So they did RNA therapy, they injected an infant with an RNA therapeutic that started in the lab studying bacteria. And so that's kind of what we do.
And I will also, this is Kate Bales, just thinking about the training aspect. We all come from so many different backgrounds, and that is only to our benefit. Right? I'm a trained vision scientist. I'm not sure what you guys got your PhDs in. You want to say?
Ion channel biophysics.
We're so diverse, but that allows us to bring a ton to the table. So all of our no PhD is ever alike. We're all very unique in our training and our education, but we're able, that only benefits the field. So we can take what we know and apply it to different organisms or different systems. And so, I work in a unique realm of basic translational research. So what we're doing is we're taking our animal models, we're exercising them on the treadmill or a running wheel, as a voluntary exercise, for instance. And so then we're able to take that and translate that directly to a clinical trial, because exercise does not require FDA approval. It is cheap and effective, and anybody can do it within their own, what you can do with yourself, with your mobility. And so, but that's a unique realm, but it does require rigorous, and I will say basic research is just like any other service. It is very intense, very rigorous. In order for things to move forward, we have to make sure what we are reporting and what we are seeing is true, and a ton of statistics have to go into this, and tons of repetitions of experiments. It's not a one and done. It has to be done in multiple people's hands, have the same result. So that's another reason why it takes so long, is because we're just making sure before we ever publish something that it's validated, it's true, and then our peers review those papers. And that's a whole nother animal of getting peer-reviewed scientific journals and publishing, getting our information disseminated properly, effectively. And so this conference allows us to do some of that, but on a larger scale, it takes a ton of manpower and a lot of passion, I will say, to do this sort of research, because it's not easy.
Malia Edwards again. I'll just kind of add to that, as you mentioned, the translational work. One of the things we can do, working with models, whether they be zebrafish, mice, rats, whatever, going up to monkeys or pigs, whatever, one thing we can do in the lab is we can do a lot of the same imaging that clinicians do, But then we're able to take those eyes right away, and we can look at them in the lab under a microscope, and we can really understand what we are seeing. You know, with clinical imaging, you can get an idea of what you're seeing, but we can really look at the cellular level and understand what's happening and take a snapshot, which you can't do, obviously, with humans. So that's, and then we can understand more of, are there different markers, different markers that we could use that we could take from maybe human blood or samples that we can get from humans that may help us understand what's happening in the disease. But I would say, you know, when we get together at a meeting like this, it is inspirational for us to interact with patients and be reminded why we're doing what we're doing, but also just interact as scientists and learn more.
Thank you very much. Just ordering
Joe?
Oh, sorry, yeah.
Yeah, Joe Porter. I do discovery research, as John says, and I would just like to agree with what everybody else said.
There's another thing, I don't know if this will maybe another thing. My lab is really involved in generating IPs, intellectual property. All that intellectual property is kind of so you come up with these discoveries, then universities are really interested in getting IP because that's how universities can make money off the farm. And so we have some of this in the lab. And so who's making the money? It's not the scientists. And then so recently, for instance, we licensed IP companies, and it's amazing how the money goes higher the farther away it gets away from us. So, like some of our ideas that we had are deploying. So it started with NIH funding. We got these ideas, and we licensed it to a company. Those ideas now are deploying 50 people in biotech companies, right? And so that's kind of where the return on the investment comes for just economic standpoint. And then that's how it gets into being a therapeutic.
I'm not Joe Porter, I'm Mary Porter. And just coincidentally, he is my nephew, so I have to call you Dr. Porter, I will. I'm also an IP attorney, so send the money over. Bring the truck, open it up, put it out here. I know what to do with it. I want to drill down. I love what Jess asked and the opportunity for the people here that never touch science to get a sense of what it means to be working in a discovery lab, thank you, John, in this kind of work. And it's companies that are out there to make money don't really do this because it's so uncertain and it's so labor intensive and so demanding in ways that aren't related to business. So thank goodness for the universities and the institutions that support this discovery research. We would be nowhere at CRF without it. I mean, it's really important. But I want to go back to the presentations yesterday, and specifically on RNA technologies. So Dr. Mack, Joe, John, the two RNA technologies that were presented, and one is the nonsense mutation, which in CRF is maybe a third of us, right? And then the other was, Dr. Mack, I'm going to be a little bit stupid here. Was it a missense or was it a...
It's an intronic variant, so it's within the coding sequence. Intronic.
Give me a nice word. Intronic is the right one?
Yeah, intronic.
Okay. And I want to go back to that and see if we can give this group a little more common language understanding of those two programs, where they might go, and where they sit today, which is in the discovery phase pretty much, a little bit, maybe a little beyond discovery, but with respect to CHM and the discovery phase. And I like to think of the, anybody have a coat with an inner lining where the inner lining gets zipped in and then you have the outer lining of your coat? When I think about DNA, I think about that as sort of like being the outer zipper. Am I going to be, and then when you get to that inner zipper, you have to zip it into the outer thing. Somewhere in there is like how the RNA
once you got your DNA in place, the RNA is zipping up and unzipping, and that zipping is how you make a protein, okay? I like to think of it in a visual way when I tell people who aren't scientists, this is what we're doing. And then when you have a nonsense mutation, the zipper is stuck, okay? So you can't make a protein. Just try to think about it that way. So my question is, we heard earlier about this RNA has to get into the cells in your retina, and that's quite a job. And delivery, you heard that delivery is really an issue? Because I'm going to ask a question, I'm leading up to this, the whole thing. So let's say, let's say everything goes perfectly, right? In your research, which never happens. And if it does, you're doing something wrong. So let's say it goes perfectly. These people would like to know, when can I have an injection, when can I take a pill? And what are the so let's say it's really good. And let's say we've heard about inflammation when you have a viral vector and AAV, like we've used for some of the DNA genetic therapy trials. So do we have decent alternatives where maybe inflammation might look different? Can we use, and getting back to the Catholic analogy, Dr. Mack, these therapies, these RNAs we looked at, are not agnostic. They are agnostic. Now, they're not. Which are they? All right. So, but I think that the questions around delivery would apply everywhere. The particular technologies, it would be only for those particular genetic types of defect. So anyway, what's the road to get us there if all goes well, and then kind of bring it into walking around knowledge of non-scientific knowledge, if we could do that. Because I'm encouraged by what I heard. So that's a long question. Thank you.
There's a thing called personalized medicine, which would be specific to the individual mutation that you have. And then there's a disease or a mutation agnostic approach, which is also called a platform therapeutic, which should apply to the majority of the mutations that everybody has. And so the ASO splicing mutation is a personalized approach that's directed towards the exact sequence that has the misstep. And then the tRNA approach is a platform where it treats all the nonsense mutations equally. You should be able to restore protein expression. And so I would say that if everything went well, and you did you've injected animals, non-human primates, it was safe. The fastest I think you could ever get anything into a person, and this would be recorded like 5 to 7 years, would be it could be incredibly fast from when you've done these studies in animals. And that's with accelerated approval, where the response that the individuals have would say that it would be inhumane for a person with the disease not to get therapy. And then it can take much, much longer than that, sometimes 3 times as long, to get something into a patient.
That's what I got it. Yeah, my voice is loud enough, everybody can hear me.
Channel your inner J.R. Rizzo.
You say that again. I'm still right here. Oh. I didn't hold it too close. I'll blow the speakers. Question for everybody. CRISPR. Can we talk about CRISPR, please? And how it would relate to possibility of using it, of course, for our disease.
You brought it up.
So, what exactly do you want to know about CRISPR? So, I can go on. We can talk all night about CRISPR. I love CRISPR. Do you want to know how basically it works?
No. I've seen the videos and the stuff on television, but okay, what steps do we have to do to possibility of CRISPR being used to correct the vectors or the viruses in our retinas? So CRISPR is editing the genome, right? So you're changing the DNA
which then is transcribed into RNA, which is then translated into protein. And Joe, I think, did a nice job explaining that yesterday. And so what people have up to now have done is they've delivered it by virus. So you're familiar with AAV, adeno-associated viruses. You deliver the machinery to the cells by using the virus. And I think that why viruses are still a very viable approach to this, because nature has figured out the best way to get DNA or RNA into cells. There are other ways to do this. And so, in an example I talked about, which was published about this infant, it was what's called a lipid nanoparticle. So it's a little vesicle that you make in a lab, and inside these little vesicles is a cargo, and it's generally, in this approach, to be RNA. And so that can be injected into the eye. The vesicles fuse with the cells that are important, so the retina cells, and then it dumps its payload into them, and it will then, you can then edit the genome. So this is what was done in that child with the liver disease. And I would say that because that was so successful, and there are other approaches, there are other ongoing trials with other diseases where it's been very successful, that it lowers the burden for other diseases to also have this done. And that mostly is because of safety. Because if you're editing the genome, you want to make sure that you're only editing where you want to edit and not everywhere else. And so people are getting very, very good at this. And the more data we have and the more wins we have, success, that the burden for getting it through FDA will come down. And it likely, there's a lot of conversation about, do you have to do it for every mutation and go all the way through clinical trials with a different mutation? And there's a lot of discussion about that not being the case. It's not a promise.
This is Rachel, and we had a trial at Mass Eye, a multi-site trial using CRISPR for a form of labor congenital amaurosis. And CRISPR and genome editing was a really attractive tool in that setting because the specific gene is massively large and too big to put in an AAV. And it was a gene where there's a mutational hotspot, or there's a specific mutation that a lot of people have. So if you're thinking about what's an efficient way to try to deliver effective therapy, you pick this particular mutation and you try to edit it. That said, I think we have to pick the tool that's right for the need. And I think that the mutation specificity of CRISPR is a real issue. Even if FDA approves like platform technologies and you can just swap in, what you need for that specific mutation, I were thinking about what's the most effective way to get a therapy to the most people possible as quickly as possible. It would be, frankly, what Spark and Biogen did, using gene replacement therapy. Whether it's with an AAV like they did, or maybe at some point in the future we'll be better at getting lipid nanoparticles into the retina. So I think we all need to recognize and develop all the tools we can, but we still have to figure out why the Spark and Biogen trials had the outcomes they did. And so that comes back to the work that Dr. Alleman talked about this morning. It comes back to the work that Professor McLaren talked about. Because until we understand that, and until we understand when to intervene, I think we run the risk of hitting the same endpoint for any kind of genetic targeted therapy. And that's my opinion, right? I'm not sure I'm correct, but I think it's really important to figure out when do we need to act.
My name is Angela Ricketts. I have a 14-year-old son, and I just want to say, you mentioned that this group, our foundation, is your motivation to continue to do what you do. You are our motivation. And I want to share this. Yes, they need a huge clap. Yes. 7 years ago, when we were in Dallas, I left, and my little boy was, I guess, 8 at the time, 7. He asked me when I left, he's like, "Mom, can these doctors fix me?" And I told him no. I said, "Because you are not broken. But every day, these doctors, researchers, and everyone else wakes up and they are motivated to go in and find these cures. So I want to thank each and every one of you for doing it for years on end. And maybe you don't have the outcome, but we are getting closer. And I also want to thank everyone in this room for showing up for yourself, showing up for your family, and showing up with some hope. Because when I hear men in this room tell me it might be in my son's lifetime, I want to thank you. You are our hope, everyone in this room. Thank you.